Hello, good afternoon, everyone. So we'll start because it's 2:45 and we will try to stay on time. Do I need to read the housekeeping notes? It's not your first session. I guess you knows everything by now, but please silence your phones. And you can photograph us if you want to. And so all other criteria as for the previous sessions apply here as well. So today we're going to talk about animal imaging, and I know animal imaging is a relatively new addition to the ABRF. Myself, my first ABRF meeting was last year, thanks to Dr. Gordon Robley, who was organizing last year the animal imaging session. So we're going to talk today about what does it take to put together an animal imaging core or animal imaging shared resources. So my name is Natalie Serkova, and I'm a director of animal imaging shared resources at the University of Colorado Anschutz Medical Campus. We are P30 Cancer Center CCSG supported shared resources. My co-chair is Jenna Steiner. She's actually a manager of our animal imaging facility, and her background is veterinary medicine. My background is MR physics, so I'm an MRI person. And we will be talking about what does it take to build together the successful animal imaging facility, what does it take to produce the meaningful data, what kind of data you need, and how you actually analyze this data. Can you do all analysis yourself? If so, what kind of software you need, what kind of equipment do you need, and do you need to reach out to your bioinformatic people to do something more advanced, such as artificial intelligence? Nowadays, it's everything about the AI. So this is what this session is going to be about. And we will talk about the equipment requirements, because obviously animal imaging is one of the most expensive technological cores. Probably only cryo-EM will beat 9.4 Tesla MRI or PET MRI in the costs and service contracts. We will talk about the data requirements, what kind of data, what kind of modalities you're going to need, and we'll talk about the team requirements, because it's really very unique, broad scope expertise which you would like to bring to your core facility. We will also then we will give the mics to our speakers. We have two speakers which Jenna will introduce in a bit, and they will be talking about a scientist perspective, academic perspective on identifying image-based biomarkers with Chet Haney, and we will talk about also, okay, I got the data, what do I know next? And this will be an industry perspective by Mohammed Farhoud. Just a little bit. So I started in Colorado back in 2003 from ground zero. We didn't have any single animal scanner in Colorado at that time. And it took me approximately $5 million of the federal grants, 8 S10, and probably equal amount of institutional support to bring to our facility everything, starting with 3 IVIS spectrums. You see it here. Let me see if I can use yeah, I can. IVIS spectrums, and also we have one IVIS spectrum CT, which is now Revity. We have a 2 CT scanner. One is a Quantum Revity CT, and one is Siemens Invion CT. We do have a PET CT, we have a SPECT CT, we have a 2 F2 ultrasounds, Vivo ultrasounds, and we have our JAM. It's 9.4 Tesla Bruker MRI. It's JAM because I'm an MRI person, and it's also because it's the most expensive. And we also have Tyler here oversees our image-guided irradiator, so it's a BLI and CT guided animal irradiator. We don't have it, but you can also get, like, for example, photoacoustic. And based on my experience, as I said, I started almost 12 years ago from ground zero. Average investment into an animal imaging facility is $12 million just in equipment without FTE and without service contracts. So what is animal imaging team requirements? Ideally, you will have a different expertise, such as veterinary expertise, because you're obviously dealing with the animals. You also will be great to have a director who has an expertise in imaging, in physics, I would say, MR physics, medical physics, CT physics, and this is a person who oversees pretty much each upper level of the operation of the animal imaging core. We do have, and most of the cores have a manager, which is usually a master or bachelor level, or maybe veterinarian technologist. They do oversight of the daily operation. So director writes the grant. This is a person who writes S10, writes reports to the, sometimes even grants from our major users, our P30 reports, and so on and so on. Jenna is the person who is to-go person for everything and anything on a daily basis. Also, she oversees the training of the users, and also iLab billing, and so on and so on. We do have several technicians. I would say ideally probably have 3 animal imaging techs, ideally with a different background, maybe once again physics, maybe analysis, maybe a vet tech, and then you also could have a technical director, which we don't have it yet, but we probably will be introducing more for like QC, QA, new protocol development. And the question is here, do you need an image analyst in the core, or you can maybe use an image analyst in your biostatistic bioinformatic core? So this is something for the discussion. And honestly, my postdoctoral and graduate students, they're not really involved in the core operation. They just do they're using the core, but they're using the core for their own projects. And in addition to all of this, we also have so-called administrative operational team, such as internal advisory board, our sometimes major users and leaders in the, from the university. Here we just write who they are. And we also work very closely with our financial administrator, cost study every year, our reports, iLab billing, and we also work, we are behind the barrier. We were one of the first facilities in the country to build animal imaging in the vivarium. And the great thing about this, we have like 150 veterinarian technologists in the OLAR, Office for Laboratory Animal Research, 5 DVMs who help us with anything and everything if we need something very sophisticated. I'm an MR physicist. You don't want me to touch any mouse for the infusion of the gadolinium during the dynamic contrast enhanced MRI. So the data requirements. We actually have created our data management and storage policy, and for each and every study, we're looking for image data sets. We have a DICOM or text or PNG or TIF files, which we share with our users at the day of the imaging. We also provide them either with image segmentation histograms ourselves, or we teach them how to do it. By the way, we do have like a reading room for our users to book the time on the workstation, which have a different software, and we'll talk a little bit more about the software, which are very useful. We also provide them with quantitative image analysis, such as signal intensity, tumor volume, flux, SUV, for PET, ADC, for diffusion-weighted imaging, and so on and so on. Usually they don't like to do it themselves, except of the IVIS, MRI, CT, they prefer us to do it, and we still have expertise to do that. And then, if you want to get very fancy, you can start applying MATLAB and Python. This is where sometimes my students come into question. Or you can do deep learning and AI. For this, you need somebody with a strong bioinformatic background. Either hiring yourself, maybe paying partial FTE from the other course, such as bioinformatic core. This is a question. And this is just if you're interested, when NIH introduced data management policy, we actually provide this guidance for our users, how to report in their R01 what the animal imaging does for SOPs for data management. Happy to share if anyone needs it. And I will stop here with my introduction, and Jenna is going to introduce our speakers.
Thank you, Dr. Serkova. Our first speaker will be Dr. Chad Haney. He is a research professor in the Chemistry Life Processes Institute and the Departments of Radiology and Biomedical Engineering at Northwestern University. Dr. Haney has a PhD in biomedical engineering and has been working on multimodality imaging for nearly 2 decades. His personal research interest is attempting to identify image-based biomarkers as a signature for response to chemotherapy. He is well-versed in multiple imaging modalities with particular expertise in dynamic contrast-enhanced magnetic resonance imaging. Although cancer research is his primary area of interest, he has collaborated with a diverse group of researchers from evolutionary biologists to material scientists. Running a preclinical imaging core involves incorporating preclinical imaging into disease models outside of cancer, such as muscular disease and Alzheimer's disease. Our second speaker will be Mohammed Farhoud, and he is a founding member of Exodus Imaging. He holds a master's degree in biotechnology from the University of Wisconsin-Madison and is currently pursuing his MBA at Arizona State University. As a scientist specializing in medical imaging, Mohammed takes an application-driven approach to the development, commercialization, and support of medical imaging devices. An entrepreneur at heart, he is inspired by the scientific community to create innovative devices that enhance and complement best practices in molecular imaging. And now I will introduce Dr. Haney to the stand. Thank you.
Well, thank you for the kind introduction, and thank you to the ABRF for inviting me to present today. I went to a regional ABRF meeting many moons ago, and when I introduced myself and said I was involved with imaging, the other person would often say, "Oh, light sheet or fluorescence?" No, small animal imaging, different imaging. And I'm just pleased that now animal imaging is a session, and it's becoming a bigger part of ABRF. So thanks for that as well. So a quick outline. I'll touch on the background a little bit. Natalie covered some of the background, so maybe I can go a little faster on that. And then I'll ask the question, why would you want to do quantitative imaging? What are you measuring, and what are the endpoints that relate to image-based biomarkers? And then hopefully get through some examples. So Natalie already talked about the different modalities, but I'll just point out that basically what you see there, other than the optical imaging, is the small animal version that you would see in a hospital. And what that does for us is it automatically makes us translational. Just to orient you as far as scale, let me see if I can get the pointer to work here. So we're in this range, so maybe 50 micron up to millimeters. That's kind of the range we're at. So whole animal, organs, and then slightly less than whole organs. This is an interesting comparison that came out. Although it's in 2010, it's still relevant. I summarized it here. I'm just gonna go through quick highlights so I can get to the examples which I think are more important. So for X-ray computed tomography or CAT scans that you might hear in the news, let's see here. The main thing to point out, it has very high spatial resolution. It's often isotropic, which means the same in each direction. It's expensive, and it does have a little bit of radiation. MRI, which is our favorite, is much better soft tissue contrast. Even without contrast agents, you can do functional imaging and anatomic imaging. With CT, you're doing pretty much just anatomic imaging. It's very expensive, and it requires a lot of skill. For nuclear imaging, obviously it involves radiation. You have to give a radionuclide. The sensitivity is orders of magnitude better than CT and MRI. It has lower spatial resolution, but you're really winning on the high sensitivity and also the ability to image biological processes directly. I'm going to skip over EPR. Optical imaging, fluorescence, and bioluminescence. You have high sensitivity and the ability to do reporter genes or cell tracking, It does have low resolution and it's often just two-dimensional. And ultrasound is low resolution and it is low cost, so it has the opportunity for high throughput. Optical imaging is also high throughput. This is just a scale to kind of orient you as far as the comparison of spatial resolution versus sensitivity. This is a clinical scale, so if you just divide by 10, that'll give you a rough idea where it's at for the preclinical imaging or preclinical scanners. So why would you want to do quantitative imaging? Obviously, for a longitudinal study, for a single time point cross-sectional investigation, that's diagnosis or tracking response to therapy. Dosimetry is becoming a big thing in molecular imaging with the radiotheranostics. And then what I'll focus mostly on is the opportunity to develop an image-based biomarker with quantitative imaging. And the question for all of these modalities, and also with respect to developing a biomarker, is, are you doing a direct measurement? So with MRI, there are direct methods and there's indirect methods. Sometimes you're imaging the effect of gadolinium on water, for example. With SPECT imaging, sometimes there's a conversion factor to get the number of counts to activity, and that conversion factor may influence whether or not it's a good biomarker or an image-based biomarker. And then for bioluminescence, there's substrate effects. So you have to give substrate for the luciferase system, and sometimes it's ATP dependent. So your biomarker may be ATP dependent and therefore not a good surrogate for whatever disease model you have. This is a slide from a white paper at a workshop that Natalie hosted in Colorado. She already talked briefly about some of the examples. So for cellularity, you can do standard uptake values from PET imaging. Diffusion-weighted MRI can give you apparent diffusion coefficient. I won't go through the whole slide, but these are just examples of endpoints for quantitative imaging. This is an interesting cartoon for cell tracking and reporter genes, but it's also interesting to use to ask the question, what are you measuring? In the first row, it's a single time point or a single injection of whatever agent you're using, and you lose signal over time. That could be because of decay, it could be due to cell division. And then this bottom panel is 4 different examples of how you can boost that signal at each time that you take the scan. So these are both examples for longitudinal studies, which again is one of the benefits of doing in vivo imaging. You get to do multiple time points rather than have to euthanize the mice at various time points or for various cohorts. So this bottom panel, you inject your substrate or you inject your radio tracer or whatever you're using, and you get more signal at that time point. In the first example here, this is a phosphorylation, this is taking advantage of phosphorylation to get the radio tracer to stay inside, so wherever it stays in the cell, you're imaging that particular cell. On the second example, you're introducing a SIM-porter that only allows that radionuclide to accumulate in the cell and therefore imaging that particular cell. The third one has a receptor, so your radionuclide would be bound to whatever ligand goes to that receptor. And the fourth example is a fluorescent protein, for example, GFP, and then bioluminescence with something like luciferase. So now I'll get on to some examples. This is a study we did using MRI, and again, this is in an effort to be translational. So the Duchenne muscular dystrophy is in young boys. So we can do MRI on these teenage boys. We can also do MRI on mice for this mouse model. And I was asked to add a touch of AI. So this is one example that has a little bit of AI in it. So we imaged the hind leg in a 9.4 Tesla MRI measuring T2, which is a characteristic from MRI, tissue property that you can measure with MRI. We used AI-assisted segmentation to speed up the processing. Then we would generate histograms of the T2 values. And the ultimate goal was to be able to triage the mice into severe versus not so severe, so that when you're doing your your drug study or your therapeutic arm, you're not accidentally including a mouse that's very mild in one of your groups and then skewing the data. Because this mouse model and also the human model is known for a wide variability in severity of disease. So this is just to orient you what the images look like. So the hind limb is kind of folded like this. These two bright spots are standards that we use to normalize the data. And this is the example of manual segmentation where we're trying to exclude fat and bone. And this is the automated version. And I'm trying to speed up here. This is what the T2 map looks like with the mask overlaid so you don't see the bone and the fat tissue. And this is the result of the different models. I didn't go over in the background, there's two mouse models. One is severe, which is the D2, and the MDX B10 is less severe. And so you can see from the histograms that these are different. You can also see by the color scale, this is the control mouse, that they're different. And what we're trying to do is get a number so that not only can you see with your eye these are different, but you can also see in the histogram that there are different peaks, they're skewed to one side in one case. So how can we turn that into a number? If we can turn that into a number, then you have hopes of turning that into an image-based biomarker. So we did some histogram analysis, and we came up with the interdecile range and the Pearson mode skewness as our two metrics for severity of disease, and ideally when we get to the treatment stage, we can look at response to therapy. So with the AI-assisted segmentation, we were able to reduce the segmentation time by 10x, and the Pearson skew and interdecile range of the muscle T2 were distributions that repeatedly indicated muscle severity. And more importantly, if I didn't mention it earlier, you always need some kind of metric that is your ground truth, whether it's histology, or in this case, it's Evans blue dye uptake, which is just a measure of leakiness. So you inject Evans blue and it leaks out when the disease is very severe. So both of these metrics correlated with Evans blue dye, which in this case was our ground truth. This is the slide to just show that it did correlate with the Evans blue dye, and this is to show that it's reproducible. The distance between the two imaging sessions was short because of disease progression, so we couldn't do, say, 2 weeks later, but if we can do one to 3 days and show that this is reproducible without large swings in either of these metrics. So what I presented before is a subset of texture analysis. Texture analysis is part of what is also called radiomics. And one of the things that's tricky about texture analysis is you get numbers, but they don't necessarily relate to something physical. But it's still useful nonetheless, especially with medical imaging and molecular imaging. So I know Mo is going to talk about some software options. I just wanted to mention that the texture analysis from the previous screen is available on GitHub from the Memorial Sloan Kettering group. It's called SIR. So I have time for a few more examples. This is switching from MRI to SPECT imaging, which requires a gamma emitter, in this case technetium. So this investigator or collaborator developed a radio tracer that binds to the cell membrane after it's damaged, which typically inverts, exposing some of these like phosphatidylcholine, phosphatidylethylene, and things like that. And what he's trying to develop is a signature for response to therapy or tissue damage. And you could eventually use AI or machine learning to take, this is a graph of the signal from each organ. Unfortunately, you can't read that. These are individual organs along the bottom, and this, the y-axis is the signal you get from the imaging, which we also confirm with gamma counter. So we harvest the organs and compare the image with gamma counting. So you could take this and develop a toxic profile for each drug. And the idea is eventually you could do personalized imaging, because you could do the same technique in humans. So for each patient, you could develop this data set and say, okay, for you, we need to lower the drug because it's affecting your kidneys, and then maybe for another patient, the dose could go higher because they're tolerating it better, and you just kind of titrate it for each patient. And again, I said earlier, you have to have ground truth. So here is cisplatin and control, H&E and tunnel staining, and this is what a typical image looks like for a healthy control and the kidneys in the rat that was treated with cisplatin. Moving on, same technology, but now applied to cardiovascular disease. So in cardiovascular disease, what they typically use now is a gadolinium in what's called delayed enhancement. So they'll inject gadolinium, take an MRI scan, and just look where it accumulates over time. The problem with that technique is it doesn't show you the area at risk. It only shows you where the tissue is damaged. So the goal with this project is to be able to triage patients that have damaged tissue but not dead tissue, because then they're a candidate for bypass surgery. If you do delayed enhancement, you miss the opportunity to see how much of the tissue is damaged but not dead, and that patient would be a candidate for surgery or for bypass surgery. So this is what a SPECT scan looks like of a rat heart that's had infarct in our model, and then this is the staining, this is the SPECT image, this is the staining, and then more slices down below. Again, ground truth to show that our biomarker is recapitulated in the H&E and tunnel staining. This is another SPECT example. This one's not really meant for an image-based biomarker, but it's meant to visualize and quantitate kidney function. So this collaborator developed a peptide that he can change the size to measure kidney function, and we add a radio tracer so that we can see it in mice. So he's had various models to show kidney damage, and the images match what you see in the histology. So in summary, some of the endpoints that I showed you can use for biomarkers. You need to understand where they come from. In some cases, it may be a surrogate, it may be a direct measurement. You always have to have ground truth to see, does the biomarker match, or does the image match what you would measure with traditional methods, understanding whether it's direct or indirect. I didn't really touch on the correct statistical methods other than with the histograms that we're doing, but we've seen other examples where if you use the wrong statistics, you get the wrong answer. And so that's important to have a biostatistician on board when you're trying to develop these biomarkers. And I'll just say that pretty pictures can be quantitative. And with that, are we taking questions now or a little late?
No, we just will go with the second talk, and then we'll have 15 minutes panel for the all questions, if it's okay with everyone.
All right.
Thank you, Chad.
So it's always horrible going after these speakers because, like, your expectations are pretty high now. So just lower the expectations as we get started. Thank you, Natalie and Jenna, for inviting me to talk. I actually came to this meeting years and years ago. My background is heavily in nuclear medicine and MRI. I actually did a stint in digital pathology and block face imaging. So it was I went from imaging humans to mice, down to tissues, and then now I'm back in nuclear medicine again. So you've got the data. What are you going to do with it now? So I'm currently a founding member of a PET-CT company, so we do preclinical imaging on in vivo animals. For a stint, and I still do it on the side, I actually took on analyzing data for pharma groups. So there was and like some academic partners, because people were like, hey, especially during COVID, people were like, I need a figure for my grant. I don't have any postdocs. I don't have any students. So I saw a business opportunity, so I actually started contracting myself out to create figures for grants and papers and things like that. Worked out pretty well. And then I decided to go back to school. So I don't recommend going back to school in my old age, but it's fun. So some of my examples, so I used to be the core manager at the University of Wisconsin, helped bring in quite a few pieces of instrumentation, do a lot of training with things like that. In my past, I've been at Siemens Healthcare. I've built cores in CROs and pharma groups. I've been commercializing and creating products. I've taken products from an R&D bench and actually taking them through approvals and clearances and got them into people's hands and actually helped you get that paper, that first paper, get that first instrument from the company and get that first paper out there is always, you know, it's the crowning achievement for the company as I see it. We'll go this way. So we're going to focus on preclinical, but as Chad alluded to, everything comes from the clinic. So our decision-making process and things like that are like, why are we using these models? Because we want to understand human disease. We want to understand things like that. But we have scaling issues. So anyone here do in vivo? Who's doing like live animal? And then everyone else doing microscopy? Who's doing both? So yeah, so, but it's, at the end of the day, so we did a survey once when I was at AACR, and we asked everyone, like, what's your next goal in research, right? So a vendor, and they're like, so a cellular person wants to go to an animal. The animal person wants to go to cellular, right? It's what they don't know, but they're like, I can put the drug through tail vein, I can see where it goes in my scanner, but is it really getting inside the cell? Is it just pooling there? Is oncology, is it passive targeting? Is it active targeting? So we need all these different modalities together, but they're all different data types, they're all different sizes, they're all different resolutions and things like that. And there's a big gap between them. So we're looking at like millimeter, and then we're starting at 100 microns later. So we have a 10-fold gap. So what I'm going to talk about is what are we analyzing? What are the tools we're using to analyze? One of the key things that I run into and openly with younger investigators now becoming PIs is how to interact with vendors. So everyone goes and like they start a lab and they're like, okay, I need a scanner. I don't have any money. How do I get a scanner? Okay. I'm just going to go ask every vendor possible for a quote. And then they hate us because we're like, hey, 3 months later, did you need help? No, leave me alone. Like, okay. But also networking. I got my first job working at Siemens Healthcare because we had the first Invion, we had the first commercial PET-CT in the market, went to conferences. And then as soon as I was graduating, they're like, so we have a job for you. But it's also, and I work at other conferences and how to network and things like that, but it's also a lot of people don't know what's next. You don't know what's next for your career. You don't know what's next in the instrumentation. You don't know what's next in recruiting a postdoc or a graduate student. So, you know, some applications people in industry want to go back and finish their PhD. They want to go back and finish another degree. So just understanding these things and networking helps everyone. And then finally, who here has written S10s? So I probably participate in about 10 submissions a year. So a lot of vendors and work with that. So always, there's questions you can ask, and some people don't want to ask and things like that. We have a lot of materials already like pre-written that you can just edit to fit your needs. So it saves you a lot of time. And people are like, oh, what do you know? I'm like, well, I've submitted 10 this year. I can tell you why these 2 got accepted this, you know, why they got funded. And it's in our best interest that you get funded at the end of the day, right? It's a win-win situation for everyone. The thing I think I put on the front is really the data format that people kind of overlook in the beginning, because data's big. Like, it's, you know, a CT now is about a gigabyte of data, but if you're planning 1,000 scans a year to 5,000 scans a year, it adds up, plus all the peripheral files that go with it. A PET data could be 100 gigabytes. So if you're doing, you know, now you do 1,000 of those. So people from the microscopy are like, psh, that's nothing. But it also becomes, where is this data getting stored? So that's probably the more important part. Is it in a cloud? So if we went to MD Anderson, no USB drives, no sneaker drives as I call them. Everything's in the cloud. Same thing at like Stanford, you're sitting there, so everything goes to the cloud. So then you have terabytes going to the cloud, and then your users have to somehow pull that back down from somewhere else. Is that reasonable? Is that feasible? Do you have image analysis? How is your image analysis tools working with that different thing? Some places still have hard drives. Every graduate student has their drive, and they're going back and forth. We're seeing less and less of that just because IT policies and things like that. But some softwares are great. And it really ended up being for our solutions, when I was the core manager, we just had we had computers set aside that we had on-prem servers. It's like accessing the data right there, so that graduate students, the technicians, anyone could actually, 5 computers, all the software on there, they can come and use it as they want. Depending on, we actually didn't charge anyone for data analysis. We charged people for training. And if we had to do it ourselves. If they were doing it themselves, they can use the computers for free anytime they wanted to. So we got a lot more people scanning because they were able to get their own data faster, but then it helped generate more revenue. I'm gonna go through kind of a shotgun approach of a lot of software, kind of give you the pros and cons. There's a lot of freeware out there that's really, really powerful, and there's a lot of software that's out there that's really expensive. So, sometimes you need it, sometimes you don't. Sometimes you just need to know how big is something, what's X, Y, Z of a dimension, can I draw a region of interest? Sometimes you need modeling or some of the more sophisticated analysis that you've seen Chad show or Natalie show. I am a chair for the interest group for the World Molecular Imaging Congress. We're putting together a webinar to put some of these software packages against each other. So we've offered the companies 10-minute slots in a webinar. Why should we pay attention to your software? And it's really, specifically we're looking, is there a laser? We're looking at, the first one's gonna be Dragonfly, In Vivo Analytics, and I spelled it wrong, it's Microscape. They're newer softwares out there, they're newer approaches. I like them, they're using more AI, they're using more cloud, they're doing a little bit better at the requests that people are seeing. So you do have, Analytics is also gonna present. You have VivaQuant, which is the old tried and true for nuclear medicine, PMOD, and Paravision from Bruker, who are not, but they become costly. My favorite pathology software, QPath, anyone? QPath? All right. Z hosts a great workshop if you're in San Diego area. I highly recommend it. Fiji for CT, AMID for using nuclear medicine, 3D Slicer for looking at 3D renders. So I've been using ImageJ since I was in high school, and it keeps growing. The community's great, but it's not optimized for anything great. Like, there's plugins to optimize things. So the good and the bad is it's lightweight. You can install it on any platform. You can put it anywhere. You can train your students. But if you can code, it's great. But then if you're looking at terabytes of data and you need to open 3D volumes, you start running into issues and things like that. AMID is my go-to freeware in the nuclear medicine world. It hasn't been updated in years. Andrew's the clinician now, the guy that wrote it. But there's a 32-bit and a 64-bit version. One of the reasons I recommend this here, it actually has the ability to view dynamic data in PET. So we're watching the probe go through the animal over 90 minutes or things like that. So it's very basic ROIs, but you can visualize really quickly. It's really installed very lightly. Nothing AI, nothing complicated, but goes anywhere. 3D slicers on the CT side, it keeps getting more powerful as far as I'm concerned. It rivals some of the more expensive software tools for doing your thresholding and your ROIs. QPath, I probably pull this software up at least once a year, maybe twice a year. But every time it's impressive, tons of videos, tons of implementation and things like that on how to use it. This was my first, hadn't done pathology since the nightmares of undergrad when I had to do it. And then when I went back to digital pathology for work, started using this. Super, super powerful, super easy to use, and very intuitive as far as I'm concerned, but might also be just because I go to the workshop and because I watch the videos. Aura is an interesting software package. So it's from Spectral Instruments, who's now owned by Bruker. But their software will open data from other optical scanners as well. So if you have a PerkinElmer or Revity, which you can't open their data on anything else, the Aura software will actually open the PerkinElmer scanner. So and they've built in a lot more tools recently. So fluorescence, bioluminescence, typically 5 animals per group, because that's how many we fit in a cage. But then you're doing multiple time points or you're doing multiple sessions. It does a really good job of loading all the data together, kind of histogramming all the data for you and saying, okay, I see this animal, I see you have 5 animals, here's 5 data points, is it just overall uptake and things like that. So very clean analysis push button from the get-go for you. Living image, the tried and true of Revity, formerly PerkinElmer. So their software is very expensive, but it's also the acquisition software. You run into the negative of if you don't have multiple licenses, you're not letting someone collect data while someone wants to analyze data. They've built in tools because they have a CT now, so you can actually you can incorporate the CT data. Living image is owned by PerkinElmer, Revity now. It doesn't necessarily always play well with other vendors' data. All DICOM is not created equal, even though the NIH says it is, so just as a disclosure. Dragonfly has actually become on my radar in the last year. So very high-end CT analysis, free academic licenses that are really powerful. You can pay for support, you can pay for add-on for industry and things like that. Tons of AI tools, tons of, but lots of plugins, things like that. The person I interact with is based on the West Coast. They're a Canadian-based company, but it's a gateway. So if you're, how do I scan, I'm doing high resolution structural CT or things like that, where I've come into play is actually groups imaging samples at high resolution and needing to segment and using their segmentation tool very quickly and very easily. And it becomes, if you have someone in the lab or someone that's very into computers and things like that, the more you beef up your computer, this thing just becomes more and more powerful. So GPU driven and things like that. But I've just seen more and more of, I'm looking at this, I see more and more of our end users using it, so then I have to start investigating, should we spend the time as a scanner company making sure we play well with these software companies and things like that. But this has come on my radar surprisingly. In vivo analytics is probably the closest to a real AI segmentation in vivo company you'll see. And why that is, so I'm gonna call it the mouse sarcophagus, and they'll hate me for calling it that. It's okay in Middle Eastern, so I can use that term. So if you look here, the mouse is actually put into a specific position. So this is the limbs out, there's a limbs in one for bioluminescence and fluorescence. So there's a holder for let's say 16 to 40 grams, so every 2 grams there's a different size. So you put your animal, whether it's in optical, whether it's in nuclear medicine, you can do 5 at the same time in optical, and then it will scan them, or you put in your IVIS or your AMI or your Logo or your Sophie Exodus PET CT, goes to the cloud and segments them. Because the animal's always in the same position, because we know the weight of the animal and the relative scale, it goes through and it's the most hands-off AI application, completely cloud-based, gives you interpretation of where these organs can go. Where does it fail? Okay, I have pancreatic cancer that's pushed everything aside. I have different models, but we see a lot of use in initial drug discovery. I'm working as a chemist. I want to see, is my drug going through where I think it's going? Is it clearing correctly? And it does a really great job of incorporating all those things. It's a cloud-based solution. So this way, if you're collecting the data and your users are somewhere else, they have a login and they can go do their own work with it. This is a newer company. I spelled it correctly here, Microscape, that came out of USC. So they kind of came out during COVID. This is a subscription-based model. Probably one of the best companies I've seen recently, how they approach Z-Stack data. So multi-channel imaging for Z-Stack. So when I was working in digital pathology, we're slicing whole tissues, multi-channel, which became a very difficult issue. But now, so think autoradiography techniques, but you want to see the bright field, you want to see the white light or things like that. So now you have tools that can easily do that, but also take your CT that you did with the animal, your PET, you did your animal and MRI, and register them all together at a cloud-based solution. They even advertise it on the phone. It's really hard to, you can use it on the phone as a viewer if you're not going to analyze on your cell phone, but super powerful tools. VivoQuant is probably one of the more prominent ones to date because it's been supplied by most of the vendors. It's based nuclear medicine, so that's where it excels. So it does SPECT data and it does CT or PET data really well. Where it doesn't excel is going to be in large, high-resolution data, because PET nuclear medicine is not necessarily high resolution. So if we're looking at 10-micron data from a CT, 20-micron, you have to have the hardware behind it to support it. So if you have a 10-gigabyte data set, you need a 20 gigabytes of RAM to open the data and to actually work with it. So it's kind of, when you're looking at these software packages, what resources allocated, we supply 128 gig RAM PC with our scanners, because when I was a student, that was the worst thing ever, is I collected data and couldn't open it. So we want to make sure that people are not running into that, because otherwise you call me right away and complain and yell. But it can handle multi-data. This is one of the first software packages, everyone does this now, but this is one of the first software packages that solved the problem of how do I take the data from my CT that's at 100 microns to my PET that's at a millimeter, or to my MRI that's at 50 microns. In bringing in the data, it resamples the size. So whatever you set as your reference, your anatomical, every data point coming in, every image file coming in, has the same matrix size, has the same resolution. So it reslices on the import. So when you're running into how do I connect this modality to that modality, different resolutions, different sensitivities, this problem's been solved already. So almost all the software packages do this now. Imalytics is the newest kid on the block. So they are hardware agnostic, so just like VivoQuant. So this means no matter what vendor you're using, they're going to open the data. They're using more and more AI tools in their segmentation. So when you're drawing, for example, a kidney, or you're drawing a region of interest, you can tell it, okay, continue to fill in. It knows what shape it should be in when you know liver. So it's going to try to find those bounds. In micro CT, you may not have that great contrast. So it's using some knowledge of what the model should be like to help you segment and draw your ROIs quickly. And again, big impact for nuclear medicine is the dynamic visualization. But you can see, you can get really nice, clean ROIs fairly quickly. PMOD is this. So this is still the farm size students that uses MATLAB to do their modeling. So this is from Bruker. So if you're using PET or you're doing kinetic modeling, so anyone that's doing neurology, this is the first question they have is, can we use PMOD? Because it has the most atlases, structures, and different things like that, really becomes the point. It's expensive, it's subscription-based, lots and lots of plugins. They're now purchased by Bruker, so they're a little less friendly with other vendors, but you can use them with other scanners and things like that. Paravision, I just put it in here because we had MRI people, and I don't really use it, but it's the tool that you're going to use when you're doing your MRI evaluation. It's the best tool to use because the tools are built for exactly what you measure in MRI. You can do them in other tools, but the workflows are designed. How am I going to do my T1? How am I going to do my ADC maps and things like that? All right, so lots of software. But what are we analyzing? It's, hey, I mean, how many people have ever had, hey, I want to know this. The biologist comes and they have an animal model, but what's the endpoint? Okay, so we have a CT, we have an MRI, we have a PET, we have an IVUS, we have an AMI, we have microscopes, we have a cryostat, but what are we actually doing? So we can do structural measurements. Is the question, is something changing? Can we even see it? So if we're doing brain, we're not doing CT. If we're doing, hey, we're doing lung capacity, we can do it, right? If we're doing, so. But we can also use old tricks with new data with old tricks. So this is block face imaging of 6 fluorescently injected brains while they're in vivo. So we wanted dynamic data. There was no tools to do this in fluorescence data. So what I did is took the data format, changed it all to generic, told the software that it was actually MRI data because it's roughly the same resolution, and then applied the MRI atlases to the individual brains. So you can take all 6 brains, register them to themselves. Because this is a tool that existed in MRI, it's not a tool that existed in pathology, in block face imaging, and then just said, okay, go in, change some headers, and then you could actually get quantitative, relatively quantitative information over time for these models. So if you take one thing home, data's data. All these tools I've been going through can open a lot different types of data. Think about what you're putting into the system. If it doesn't like what you're putting it in, tell it it's something else and see if it works. The other question I get most of the time, we install a new scanner, we install a new device, and I'm gonna look at this both ways for a second. I'm the person doing the training, I'm a vendor, I'm doing the training. Okay, this is how we're gonna collect data. Now I'm the core manager. That some graduate student just showed up with a paper and said, I want this. Who's ever had that experience? That's well, why do you want that? Is your drug going to, it's a nice image, it was in a Nature paper, it was in a science paper, they want that. But why do they want that? Well, are you looking to see, did your drug or your compound hit your target? Or are you looking to see how fast it got there? Or are you looking to see, can you block it? Or so it's walking through these steps. So here's a generic example. We use an atlas-based system on the CT, and then there's 2 tumors, but it was a static image, because the question was, can I see a therapeutic response? So it's a bilateral tumor. One is negative, one is positive. Can I see a measurement? So we didn't care about how fast the drug gets there. We just want to see if there's a therapy change. Now with more radiomics, we're looking at more radiotheranostics, everyone's like, I have this really expensive radiotherapy, I have this expensive chemotherapy. Can I use a generic tracer like FDG and see, can I see a measurement? Can I see a change? So I can do the CT, see the change in size. I can use FDG, FLT, very commercially available, cheaper probes to see if you're seeing a metabolic change in your tumor. Or you can do atlas-based drugs. You can do time activity curves. So there's actually a tumor on this side as well, but this is an atlas and not a CT. So you can't see it, but we can see, did the drug actually get to the tumor and how fast did it get there versus the tumor that it's not targeting. So sometimes you'll see now we're looking at, do we have active targeting, do we have passive targeting? Is it staying, so does it stay in the tumor or did it hit the tumor and come back out because it was only just leaky vessels going through? So it becomes, what's the question? Your fun fact of the day, anyone here see Ratatouille? So the algorithm used to limit where the restrictions of how this atlas can move uses the same open source code that Ratatouille was used for. So, I mean, we use lots of tools that are out there, right? And we're seeing more, I mean, you always see multimodality. So this is MRI and SPECT, so definitely not in the same instrumentation, but you just move the mouse over to a different scanner, you can register them, different modalities. The question also becomes, when you're looking at your regions of interest, so, does your ADC map your hotspots? So where you're seeing signal in your SPECT, where you're seeing signal in your MRI, is this vessels, is this dead tissue, is this pooling, is things like that. So now you can have functional MRI, not fMRI, but functional information from the anatomical MRI, as well as your functional information from the nuclear medicine, and you can go through them. So we see more and more systems that are, you need all of them chained together and things like that. That's, as a vendor, to be frank, that's the function of the S10 did that, because the S10 said you can only get one gantry, so how many different cameras can we put in one gantry? Analysis, you don't need them. You can move the scanner, and frankly, it goes a lot better with different. We'll skip that guy. This is an example we did for Stanford where we did block-face imaging. On one system, we used an MRI, Bruker MRI, then we used Siemens Invion PET CT. 3 different resolutions, 3 different modalities, 3 different data types, and put them all together. You know what the common thing was here? The skull. I can see the skull in every modality. So I was able to take the data and have this software going through and use mutual information of the skull, just put landmarks on them and have it put it together. And it was really interesting because I actually thought I failed because the drug wasn't going where I thought it was supposed to be going, but it wasn't supposed to target a tumor. It was only supposed to target the inflammation of the tumor. So it's actually working, but I wouldn't have known that without being able to put all these things together. Vendor relations, we're here to help. Sorry, we got 30 seconds. But it's useful for us to know, are you buying instrumentation, are you replacing instrumentation, or adding instrumentation? Because again, we have the text for you that we've seen succeed at S10s or internal grants or things like that. So we can lighten the load and work. And bigger companies will actually have people that will write grants with you. So things that renew, so service contracts, very expensive. Think about it, you're doing this. So make it a win-win situation for everyone. Like if you're buying a service contract, but you can't afford the top one, or you can't afford anything, or I get this one a lot. Can you sponsor my conference, or can you sponsor my students and things like that? It's almost always a yes. So I don't know if any of you guys have ever tried that or approached the vendors that you just paid $30,000, $80,000 for a service contract and said, hey, I'm presenting your data here. Can you help with a flight? Can you help send my students and things like that? If you talk to a salesperson, I'd be very amazed if they ever said no. And then the quote structure, there's recipes, how to break it out. So it's really important. Is it internal? Is it external? Is it S10? I've put together grants with 13 different departments buying an optical scanner. So the quote had to be split up into 13 different items so we can show we'll do it, it's painful, but if you share that information of listen, it's 5 departments, this group can only buy software, this group can only buy this, it helps you guys on pushing it through, because it shows the allocation for each department, and it helps us to make a sale. Magic recipe, Natalie touched on it. All right, minute over.
Chad, if you can come here. So, yeah, thank you. I think it was great science review of the software and also some business recommendations here for sure. This is now open for the discussion and for the questions. Please go ahead, please use the microphone.
If you don't ask us questions, we're going to ask you guys random questions. Just to be
Hi, I'm Ken Grice, University of Cincinnati. I'm curious about managing the life cycle. You have multiple instruments. They last for quite a long
10
time.
years.
But you start getting to the end date and you're looking at $1 million, $2 million to buy a multimodal system. How do you manage that? Because we're in that situation right now.
I will start with our experience. So the first thing, if you're planning to submit an S10 grant to replace the equipment, never ever wait until it is dead, right? Because the NIH, the reviewers hate it. I'm reviewing those grants itself. So start well in advance, get the quote from the vendor saying that the service contract will be discontinued, count how many days the equipment was down because it's old, and just provided the justification. So if it's not an NIH equipment, I will always depreciate. I will always depreciate the amount of the equipment over the 10 years. And I will have hopefully enough money for this. If it's an NIH equipment, you cannot do that. Meaning that something I buy from internal funds, from fundraising, I depreciate and can buy new, just having my depreciation fund. We also have internal, it's called Academic Enrichment Fund from the Dean Office. I can apply, but they really like if we have a depreciation to match the funds. As I said, you cannot do it with the S10. Chad, would you like to add anything from Northwestern? Northwestern, so we are semi-centralized course structure. We are very centralized with the P30 Cancer Center grant. Northwestern is an ideal formula for being a centralized course structure.
Yeah, we're also in a cancer center at CCSG. We're actually at this year or next year's end of life for the electronics and our 9.4 Tesla MRI. So we're actually going through this right now, where it still works, but it's end of life as far as the service. So yeah, so it's actually end of life right now, and we're working on an S10 just for the electronics. The magnet itself is okay. We've got other equipment. So I guess I should back up. One of the neat things about my center is it was pretty much all put together at the same time, and it's one of the few centers where each modality is within eyesight of the other. So I like doing multimodality imaging. It's easier when you can take the mouse from one room and go next door. When I was at University of Chicago, the MRI scanner was in another building, so I had to run through the tunnel with an anesthetized mouse to do multimodality. That's not fun. But the downside is it was all bought roughly at the same time, so everything is depreciating and end of life at the same time. And I don't think we're going to get a donor to set it up, because part of the center was built as a recruitment for my boss.
And I'll tell you what not to do. So a lot of it, so especially in a lot of instrumentation, we've seen it hasn't been making enough money to support itself in a lot of cases. And to be frank, the blunt truth that I always tell people is someone new needs to be in charge on paper of that instrumentation when you're submitting it, because there needs to be a change. So when you're rewriting your grants and things like that, so I've been at institutions where the MRI has failed its entire existence. So the only way it's getting new funding was there's a new director, new administration, and a new focus for that instrumentation. It's also but failed is a relative term, because you need it to support the researchers that are there. You may not just have that business existing there, but key things. Okay, if you need a new one, and if you have time, start. But we always joke, we take the coffee cup around and go ask for money from everyone, like 5 here, things like that, and memorandums of understanding from different groups. Okay, if someone knows radiology is going to start recruiting, if you don't have a preclinical PET CT or MRI, you're not going to be able to recruit better people. So you know that in the next year, you're going to have this instrumentation as a recruiting tool. Or, they have money to recruit professors and things like that. But it's the business model needs to be there, but, you can sit there we acknowledge hey, we're going to grow the business to succeed with the new one. We're not just going to keep doing the same thing.
And I don't think there's a recipe just to, sometimes you have an instrument, if you don't have enough publication on existing instrument, you cannot go with the S10. If you don't have a history of 5 to 12 R1s on this instrument, you might do a lot of industry, but you cannot go for the S10. And you can just be, once again, institutional support. My first PET CT, because I knew that I don't have any chance to get S10. I'm not a PET CT person. Colorado was never had anything. I went for the fundraising. Actually, I reached who likes ice hockey. I went to Avalanche team and said, would you mind to sell your, everything what you have, shirts, and we did a fundraising. We were selling the Avalanche T-shirts from the players, and I raised almost $200,000 and squeezed $500,000 from my dean, and so I bought up a PET CT. But yeah, so there's different recipes, right? I would still try to do S10 no matter what. I never done MRI NSF because we are too biomedical. Yeah, but it definitely, you can, and there was actually a session about philanthropy, right, for the core.
Yeah, you needed to be in that one.
Yeah. Hi, Jennifer.
I've seen a lot of R01 funds, leftover R01 funds, fund instrumentation, because they have essentially the consumable portion of the R01 became a part of the money that went into the scanner, because then your depreciation, the scanner itself became the consumable for that researcher. That's it's only going to last 5 years, so this is how I'm going to depreciate that and consume that. So there's be creative and talk to your program,
Officer, yeah.
It's there's the recipe, but there's other ways of going at the same recipe. But being creative is very fruitful a lot of the times.
Any other questions? Actually, I have a question to Mo. You did mention analyze, and we're using analyze quite a lot for the CT, especially bone analysis. Do you have any comments?
Yeah, so it was, I had 20 different groups. So analyze is,
Mayo.
what?
Mayo.
It's Mayo Clinic. It's actually most of the tools that are built in the spectrum software based off of Analyze. But I put in Dragonfly because I've seen more people switch to Dragonfly
Okay.
from
And
Analyze.
it's free, right? Because,
So there's a free version of it.
It's a free academic
Yeah.
Okay.
license, but if you want support, it's $5,000.
Oh, That's what
a
analyzes.
year?
Yeah.
Is- So it's the same as it?
Yeah, Analyze is,
Okay.
yeah, it's for the like costing, it's almost the same now. And Dragonfly seems to still be doing more updates.
Yeah, and I also say that my question is always, do you hire a person who has a all, AI and bioinformatic background? It is expensive, they're very expensive, so I think my take in on this would be, we do have a biomedical informatic department, our dean just funded it, so I would just rather go there and do on the collaborative or just partial support of the FTE for one of the analysts, because to hire a person into the animal imaging core is just too expensive. I don't think it's substantial. You can just,
I think so if you're gonna hire someone that's on the analysis side,
that's fine.
you should do, but it should be someone that frankly knows more MRI than other modalities. Because it's the other stuff, if you know the MRI side of things, you know the image processing and things like that and the physics behind that, which can go into the other modalities. And it's a benefit. You can go either direction, but it's the MRI person, you're gonna have a lot more questions on the MRI analysis than drawing a region of interest in the other modalities. So it's that person can do the other modalities. Well, it's sometimes harder to explain how do you do an ABC map? Why are we doing T1, T2 contrast? Why are we doing these things?
That's basically where I started. I had an MRI background, and then the person that was running the small animal nuclear imaging left, and the faculty director said, hey, do you want to learn how to do this? And so I became the interim technical director for small animal nuclear imaging while I was still doing my MRI work. And then once I knew nuclear imaging plus MRI, then Northwestern wanted to recruit me to start their small animal
I
imaging.
think just because the guys that are out there.
Yeah.
Have you guys tried Nimbus that Revity is creating? Are you familiar with that?
Nimbus, no.
It's AI driven, and we had Ryan Gesner talk and they're trying to get any type of imaging data, I forget who they're working with, someone I think at MD Anderson with MRIs, and using the AI to be able to, detect the tumors. And this is the same group that created or developed the, the Vega ultrasound. And so trying to, for us to put imaging back to them, I just trying to work out a data transfer, and my MTAs, my lawyers are really slow at VCU, but I was just wondering if has anyone else worked with them? No, you might look it up, because right now they're looking for partnerships, and it's I'm pretty sure that's like Nimbus and Revity.
Yeah, so I looked up their stuff. I didn't, to be open, it's very, very new. So
Yes.
They are, but there wasn't enough on their website to share, but it's, I know we know the guys that that did the Vega and things like that. It's going to be, and MD Anderson's a really good group, because if you've ever been there, they have all their MRIs, and it's just they're doing as many MRIs that we do on optical, a normal lab will do on just IVUS. You just see them there might be a conveyor belt on their MRI of doing all this stuff.
Yeah.
It's going to, the work on AI is going to take a while, my bias is going to take a while, because every animal model that comes into the core is so different, and you need a large library to get there. So I think that's why I'm the most impressed with in vivo analytics, because you have to put the mouse in this position.
That makes sense, yeah.
Because think about it, if it's not a healthy mouse, and if you go from a bulb C to black 6 mouse, things like that, the anatomy is slightly different. So you're sitting there, so you run into how do you characterize these things? Sometimes you don't care, because it's in the brain. I just need to look in the skull. It works. But we're seeing more and more people using cloud-based solutions from a vendor side. We're seeing more and more academics push back, not the academic researcher themselves, but the university, because now it's going to someone else's cloud off premises. So we actually require some of the people we work with, they have to have an on-prem solution, because a lot of academics, they don't want to send it to random cloud, and the academic is fine with it. If they're legal, like, well, who owns it once it's off campus, or,
Right.
yeah.
Since we mentioned the MD Anderson, I don't know if you know that Preclinical Imaging Consortium, Mo is probably one of the founder. It used to be, what is called, Midwest, right?
Yeah, it was Midwest.
Midwest, like when I first attended 2016, 2017, it was like in Vanderbilt, at the WashU, right? And then during the pandemic, it's kind of became national. So immediately after pandemic, the Preclinical Imaging Consortium 2023 was at the MSU, Michigan State. Last year it was at Colorado. And this year in April, it's going to be in MD Anderson. So since you're all animal imaging crowd, I think it's a really great one. It's only about animal imaging, like 3 days, like at the leading academic institutions. So just check 2025 Preclinical Imaging Consortium, MD Anderson organizing it, and the program looks really great. So
About almost 200 people now registered for the meeting.
yeah, so it was 156, Jenna, last year in Colorado. It's really great. I mean, if I may say so.
Everyone on this table is involved with it somehow, so
Yeah.
there's no bias.
Yeah, I went to the first one at the Mayo Clinic.
And actually, Chad also hosted it in 2019, right? Northwestern, yeah. Yeah, it's a great one. Any other questions?
So I would just like to say, I'm so excited that we've got an animal imaging group now, because that's like been forever that we have it. So like, yay, this is super exciting. We're already starting to work on next year's program.
Yes.
There's a board out there with sticky notes. Give us ideas. I mean, if we get 50 suggestions about animal imaging sessions, then we can justify having a lot more sessions next time. If nobody says anything, it's hard for us to put that together. So any ideas you have, people would like to talk to, we're even looking at like vendor workshops, maybe somebody come in and teach us how to use one of the software packages, or, things like that. So any ideas like that, are things that would be helpful, that might be different from what you're getting at other conferences, please let us know, because that would be huge in helping build this community.
Great, thank you. Yes.
So one idea for next year is, at least at the Cancer Center at Northwestern, we're trying to develop a pipeline where, as Mo said earlier, the microscopy people want to move to animals, and the animal people want to go down to microscopy. And so we're working in the Cancer Center with someone in biostatistics, someone from the microscopy core, and myself. And we're trying to develop a pipeline so that before you start your study, all three of us will come together and say, okay, you need to talk to the biostatistician, but you also need to talk to the microscopy person to make sure you understand what's going to happen after we're done imaging, and vice versa. So this pipeline has all the three core directors involved, and that might be something to entice more than just the small animal imaging people to attend a session like that.
You're missing somebody. Histology.
Histology.
Well, I skipped over that to the microscopy, but yes, we can make it 4 people instead of 3.
Actually, honestly, maybe Gordon can add to this. To find an animal pathologist, it's very difficult. And yes, yay. Right, it's so difficult. So in Colorado, fortunately, we have Colorado State University. We often send, because we have sometimes troubles to do like reading the animal pathology with our human pathology department.
We have a pathology, animal pathology is a very small field. Everybody else is, and it's very insular, and academic, like animal model animal pathology is a subset of that. So we do have a comparative pathologist consortium, and there are conversations happening about trying to bring that group a little bit more together with ABRF, so that there's a resource there.
And like, I don't know if you went yesterday to the CCSG, like Cancer Center, they talking about having like national course with very specific, unique expertise. We might not need each P30 cancer center to have veterinary pathologists, but if we have, right, absolutely, but if we know that there is, for example, I'm not sure where you're from.
I'm University of Michigan.
University of Michigan has it, so yeah, just to have a consortium and send the samples there, this would be phenomenal.
Well, you need them to be able to prep the samples correctly.
Well, yes, yeah. Yeah. Yes,
Yeah.
yeah, it's great,
Yeah,
wonderful.
I think that's the biggest thing, like I've been trying to work on that, educating my groups, because if they don't, that's why I said that, Chad, right? Because if they don't have it fixed right, then the rest of it, you can't
Right.
fix their fixation problem, right?
Yeah.
And I think that's what we have to make sure we're doing that work, and that can start with all of us doing the animal work too, telling them, hey, have you talked to the histologist or your pathologist?
Yeah.
How you want it, so we have to remind them too.
Yeah, so we always did, we defined the endpoint, like, when, you know, we always have the meeting with the new user and things like that. We defined the endpoint, and I'd always put in like, you know, we can't do these things with this endpoint, the way the study is designed. Because the number of times they come back, well, I can't do that with my tissue now, or I can't, like.
Yeah,
things
you
like
have,
that, like-
yeah, if you have a tissue, it's too late, right, to do at new endpoints, for sure, yeah.
I really like where you said the first question, I can't remember which speaker said it, but asking them, well, what is, you bring a shiny paper, and what is
Yeah.
the question you're trying to answer? Like, I ask people that every day, all day long. And I think it was really beneficial for me as a pathologist to have a session like this, because I have my area of expertise, but sometimes people have a misconception of what the histology will tell you, and they'll come, I'll talk to them, they'll tell me their question, and I'm thinking this is really not a histology question. So whatever your field of expertise, MRI, CT, pathology, you have to have at least a passing acquaintance
Yeah.
with the other technologies, because sometimes I might want to direct somebody to in vivo imaging.
Oh, I've convinced people not to buy scanners from me. I've convinced people not to buy scanners from me. Because they're like literally because they came up, they saw the paper, and they're like, okay, we have a new, like the core's like, we need this. And like friends of mine, they're like, okay, we'll raise money to buy a scanner. I'm like, it's not gonna answer what you need.
Yeah.
Don't waste your time trying to get $500,000 when you can buy something for $100,000 and get the answer, or you have the equipment already. I'm a horrible salesperson.
Okay, great, thank you. And actually, another thing which to think about is it's large animals, and this will probably bring us closer to the human, because human imaging is not presented at the ABRF at all. And I understand it's, I mean, I'm in radiology, so we do humans as well. But yeah, so maybe one day we'll also be talking about the large animals, because they need human scanners. Great, thank you so much. Thank you. Thanks to our speakers.
Learning curve and analyze, like they're using it, you know, but the learning curve and analyze is so hard.
Goodness, yes.
Yeah. Hey, thank you. Are you going to see you in the Anderson?
Like on the AI journey.
So awesome.